As you know, I'm the teaching assistant of the course. My name is Geula, and I'll just go over a few things I showed last time to make sure everything is clear. Okay? So attendance is mandatory in the course. An absence of 10% of the classes or more will reduce the final score by 5 points. Okay, so take it into consideration. Grading. We have 3 home assignments. It's 30%, 10% each, and home written work, that's like a take-home exam, okay, on the topics discussed in the class. That will be at the end of the course. Okay, you can take the exam in Hebrew or in English, we don't mind. Okay, and the course is in English, as you know. And I want to urge you to ask anything if you don't understand or something wasn't clear or we use terminology that you are not familiar to. We are aware of the fact that we use a lot of terminology and sometimes we just don't notice. Okay, so if I said some word you have no idea what it is, just please ask. Okay. Okay, so the course's website is at the Moodle, and this is my email, which also is in the Moodle itself. Okay? Good. So now we can start. Any questions so far? Yes?
When are the assignments due?
Excuse me?
When are the assignments due?
When are the assignments? So it depends on the lectures, actually. So it would be sometime after Pesach will be the first one, and to the end of the semester, you'll have the last one, and somewhere in between the second. We give you about 10 days to answer, so there's no rush. And basically, we didn't have any problems with that in the previous years. Okay, so let's start. I know you're a little bit shocked from the last class. So this one is to make things a little bit clearer, talk about the basics, okay? And again, please make sure you understand everything, okay? Because we will use a lot of the terminology and a lot of the topics we're talking about today. So the course is about neurodegenerative diseases. So these are diseases where the nerves are degenerating, okay? They're dying. It affects some of the body's activities like balance, like movement, like memory, as well as others. Okay, and many of the diseases are genetic, but not necessarily all of them. And in some cases, we are not really sure how the genetics are. Okay, so we are still trying to find this out, but in some cases, the research is still a little bit behind. Okay, so sometimes the cause can be a medical condition like alcoholism, tumor, stroke, toxins, chemicals, and viruses. Okay, for example, head trauma can cause neurodegenerative diseases. And the most known example is Muhammad Ali, okay, which suffered from Parkinson's disease. And currently, unfortunately, neurodegenerative diseases are incurable. Okay, we're going step by step and we're trying to figure it out, but currently there are no cure. Okay, so some examples of neurodegenerative diseases, you've probably heard of some of them. The most known is Alzheimer's disease, which we also call AD. Okay, so just be aware of all the names. Parkinson's disease, PD, Huntington's disease, HD, ALS, which maybe you heard about the last summer they had a huge PR thing with the ice bucket. Okay, maybe you heard of it. This was to raise the awareness of ALS. And also retina degeneration, which is actually an eye illness, but the nerves are degenerating there as well. This is a picture of a mouse model. This is called Brainbow. In this model, each neuron has a different color according to its function. This is like one of the most beautiful things that were done in the science in the last few years. So a little bit about Alzheimer's disease. So Alzheimer's disease is the most common reason for dementia among people above 65. Okay, so around the age of 80, 85, about 50% of the people are affected with AD, with Alzheimer's disease. It causes loss of memory, judgment, and language, and also other things like other things that we'll talk more about later, but it affects the daily life very dramatically. Everything is clear so far? Okay, the prevalence of the disease. The prevalence is the percentage of the people affected in the disease. It's between 0.5 to 0.75%. Just an overview on some other diseases. So Parkinson's disease, and here's the picture of Muhammad Ali, which I just talked about. So this is the most common movement disorder and the second most common neurodegenerative disease. It affects between 1 and 2% of the general population above 65. And its prevalence is written right here. Now, people affected with a PD have very common characteristics. For example, their posture is something like that. Okay? They're not standing straight. They usually have tremor, which is one of the most common things you probably are aware of. And forward tilt, as I just showed. And they usually take very short steps. So a person affected with Parkinson's look like this and walk like this. Okay? Questions? Huntington's disease. So this is an inherited neurodegenerative disease. It's a genetic disease. We know how it occurs, and you'll have a home assignment about Huntington's disease, and you'll know everything about it. It usually results with movement and cognitive and also psychiatric sometimes disorders. This is how a brain of Huntington's disease looks like. There are very big holes. This is a control brain. Okay, so basically this structure, the basal ganglia, is degenerating. So we have no cells. We have holes there instead.
Dissolves?
What?
It dissolves somewhere?
It breaks down.
Okay.
Yes?
When you say inherited, you mean it's a genetic disease?
Yes. ALS. ALS is amyotrophic lateral sclerosis. It's also very known because of a lot of PR. And Stephen Hawking is one of the most famous affected people. It usually, some of the cases are genetic, but they're very low percentages. It's about 10%. And the others are what we call unknown reason. Okay, so sometimes it can be other genetic defects or some other reasons. So, but these are the numbers. And in ALS, the neurodegeneration is of motor cells, motor neurons. These are the neurons that execute movement. Okay, and that's why people affected with ALS are usually in wheelchairs. Okay, so this was a bit more specific, and now we'll talk more generally about neurodegeneration. So why is neurodegeneration became so common in the last decade? So as Sebastian told you last class, the lifespan has increased. Okay, here you see mortality of a cohort in Sweden, I think. Not sure about the, it's Sweden. And so people live longer nowadays. Okay, so people live longer, more damage is accumulated in the neurons, and neurodegeneration occurs. Okay. We have different factors that protect from neurodegenerative diseases. For example, antioxidants, which we'll talk about, and others. Here, by the way, you see a drawing of a watch. This was done by an Alzheimer's disease patient, and you see that something happened in the drawing. It doesn't really look like a watch should look like. Okay, so they start and somewhere they lose it. Okay, this is very, very characteristic of Alzheimer's. Okay, so as I told you, cumulative damages would eventually lead to neuronal cell death along the years. And other damages could occur, okay? And Sebastian already told you also about the fact that we have machines that are supposed to fix the damages, and sometimes they just don't do the job. Okay? Questions so far? Okay. Okay, so to understand neurodegenerative diseases, we have to know a little bit about the brain. So what is the brain? Here is a very classical picture of the brain. This is the cortex, this upper layer. This is the hypothalamus, this is the hippocampus, and the thalamus. These are all brain regions. I know it sounds like Chinese at the moment, but you will know more about them soon. And this is the cerebellum and the brainstem. So some numbers about the brain. So the brain weight is about 3 pounds, which is like a bit more than 1.5 kilos. It is the size of a medium cauliflower, or adult brain, of course. And we have 100 billion neurons in the adult brain. What about synapses? So synapses are the connections between neurons, and each neuron can connect to multiple other neurons. So it's not a single connection between neurons, but a more complex network. Okay, and we have something like 100 trillion synapses. Okay, what are the cells of the nervous system? So we have neurons, the basic unit of the nervous system, and we have supporting cells. These are cells that give the neurons nutrients, okay, food, sugar, water, whatever they need, okay, electrolytes. And in general, they're called glia. Glia is glue in Latin, okay? So these are the glue cells between the neurons. They hold the structure. We'll talk more about it soon. So we have different types of glia: astrocytes, oligodendrocytes, and microglia. And here's a picture of how they look like. So these are microglia. They're relatively small. This is the neuron, by the way. And this is an oligodendrocyte. It holds the structure of the neurons. You see? It holds everything in place. It has other functions as well, but we'll get there. Yes?
What did you say? That they transmit sugars?
For example, yeah. The supporting cells, the glia cells, they give the neurons. Neurons are really, really spoiled cells. They need a support. They need someone to take care of them. So who takes care of them? These are the glia cells. They bring them sugar, they bring them electrolytes, they bring them energy. Everything they need comes from glia cells. And we also have the blood-brain barrier, which we call the BBB. What's the BBB? So it's a barrier between vessels, blood vessels, capillaries, and the cells themselves. Okay? I'll talk about it soon. About the BBB? Okay, so the blood-brain barrier is a structure between blood vessels and the neurons, which blocks passage of things that are too large. So, for example, if you ingest a chemical and it would really harm the brain, this is what prevents it from getting there. Okay. So you already know something like, you saw some slide which looks very similar to this one. The human body is the structure of the human organism. And what are the levels of organization there? So the organism is the basic unit. It can be a human, a mouse, a rat, an ant, anything, a bird. Everything is an organism. Also, bacteria is also an organism, okay? And then we have systems, organs, tissues, cells, organelles, molecules, and atoms. I'll go and talk about each of them. Okay, so what is an organism? So this is an example of an organism. This is a fish. Okay, an organism, here you have a definition. It's a self-contained individual made up of organs and organ systems. Okay, so basically, this is very intuitive. Okay, questions so far? Just stop me if anything is not clear, okay? So what is a system? So here is an example, the nervous system, the topic of this course. We have the brain, which is the central organ in the system, and we have a lot of nerves that go everywhere. They're going to the hands, to the legs, to the periphery. We also have the spinal cord, which is considered as the central nervous system. Okay? And the system is a collection of organs that work together to achieve a certain function. Okay? Clear? I'm going over it relatively quickly, but I hope you understand. Okay? Because the goal of this classes and the next one, which I'll teach after Pesach, is that you understand the basics. Okay? Some of you have some background, but some don't. So don't be shy and ask. Okay. What is an organ? So an organ is a set of tissues connected by different structures, okay, in a structural unit. And the brain is an example of an organ. Which organs can you think of? Other organs? The heart, the lungs, okay. The skin, liver, yeah, great. Okay, what about other systems? Cardiovascular. What else? Reproduction system. Okay, so you got it. We have the respiratory system. We have digestive system. Okay. So what about a tissue? A tissue is a group of very similar cells, okay? They combine and they have some function they execute. For example, this is the ganglion in the nervous system, but we have tissues like skin, for example. Okay, so the skin have many different cells, and they work together to protect the human body from any external injury and infection. Now we get to the cells. So cells are the basic structural unit of any organism. It's a unit that can self-replicate. We can grow it in a culture, in Petri dishes, okay, and study various things like in this methods. Here is an example of a cell. So this is a class of neurodegenerative diseases. So this is a neuron. This is a very typical structure of a neuron. So this is where the nucleus is, the central component of the cell. And it has a very long extension. And in each of these, I'll talk about it, but each of these parts can interconnect with different neurons, okay, with other neurons. Okay, now we get inside the cell. So what's inside the cell? We have a lot of organelles. Organelles are compartments inside the cell. They are surrounded by membrane. This is something to define where they start and end. And they perform different functions. So probably some of you heard of different organelles like mitochondria right here, or a nucleus, and I'll talk a little bit about it. Okay, every organelle, everything basically is made of molecules. What are these molecules? So a molecule is an electrically neutral group of chemicals. Okay, this is how a molecule looks like, and the fact that it's neutral is really, really important. Okay, if it's chemically charged, It's not a molecule anymore. It's an ion. Yeah, so a molecule is a neutral chemical, a neutral electrically charged molecule, a subgroup, okay? While it's neutral, it is defined as a molecule. If it has some charge, it is defined as an ion. So for example, we have charged molecules in our nervous system that allow neurons to function with electrical charge. Okay, so they are a plus and minus charged, and they perform different functions. We'll get there. Okay, so each molecule is built of atoms, and we won't get more deeper than that, although it's possible, because that would be a chemistry class then. So atoms are the basic unit of matter. It's the smallest component, which is not exactly true, but we'll leave it that way. And it has chemical properties of an element. So the atom is built of a nucleus, which is charged with protons, or positively charged particles, and neutrons, which are neutral charged particles. And these atom is surrounded by electrons that basically they travel around here. Any questions? Okay. So we're not going to really talk about atoms in this class, because this would be chemistry. But just a general overview. Are you familiar with this? Okay, so I hope most of you are. This is the periodic table of elements. So it's a table which the first person to make this table was Dmitry Mendeleev. It has all, now it's been updated, and every here and then it's getting some updates, because we still find different elements that were not known previously. Usually they're not really stable, so this is why they were not discovered previously. But we have here all the elements of life. So we have hydrogen, we have helium, and we have many different other elements. Okay, so we'll get to the first most important molecule in this class. This is water. So the water molecule looks like this. It has an oxygen atom and 2 hydrogen atoms. These numbers are not really important, okay? And the take-home message here is that we are 70% made of water. Our bodies are 70% water. There are different chemical properties of the water, like anomalous properties. For example, what do you see here? Ice. What happens to this ice? It floats. Thank you. It floats on the water. So what makes the ice float on the same element? Okay, so this is water. This is water. What happens there? Yes?
It's like the atoms, like yeah.
Right, density. Density.
The
Right.
density is lower.
Right. So what happens when water becomes ice is that it gets to a very organized structure. Okay? It's organized like very, very densely. Okay? And in case of liquid water, they're not so organized. So this is what causes ice to float upon liquid water. Okay? And it's very important because since the water behaves in a, has these properties, let's say, okay, we need our bodies to be in a very constrained conditions to allow the human body to function, to live. Okay? And if something happens and you freeze or something like that, you understand that the cells are going to die. Okay. So what are the other molecules in our body? Here you see some elements. So we have oxygen, we have carbon, and hydrogen. These are the 3 most important elements, also nitrogen. These are the 4 most important elements in our body. They're about 96% of the total chemical composition. And we have some other chemicals that do certain functions. Now we're going to talk a little bit about the organism, not about atoms and molecules, but about other larger building blocks. So we have 70% water, we have ions and small molecules, and we have large molecules. What are they? They are proteins. They are nucleic acids, which are RNA and DNA. You probably heard of them. This is the genetic material. And we have carbohydrates, which are sugars. They're very big sugars. And we have lipids that are fats. Okay? This is not really important. Okay. So what are the size of these components? So atoms are really, really small. They're about 0.1 nanometer. Small molecules get to about a millimeter, depends which molecule. Lipids are larger, and then we have proteins. This is a virus. This is a component of a plant. And we have other bacteria that are larger and different cells. And then we have the fish egg, which is really, really large. You can see it without a microscope. So you get the message. Cells are really, really tiny. Why are they tiny? So we have a limited surface area to volume. Why is this important?
Because then generally the cell would be exposed to more cells in its environment.
Right, right. So if we have a lot of surface area, cells can talk to one another, and they can also transfer different things between them. Okay, for example, different chemicals. Okay, so just to make sure, the surface area of a cell is the area that surrounds the cell and makes it a unit that is different from its environment. The volume is the space inside the cells. And basically, most of the cells, almost all of them, have a large surface area to volume. We have also some cells that are really, really large, and there are different reasons for that. But generally speaking, evolutionarily, it's less favorable. Okay. Some cells are large. For example, this is an egg cell. This is a human egg cell. It's huge. It's 200 microns, which is really, really large. You can look at it in the naked eye. And this is a cell of the immune system, which is also really, really large. So what is the longest cell in the human body? Not largest, but longest. Okay, so the volume inside is really, really small. So this is the sciatic nerve. This is a nerve that goes all the way from the spine to your toe. Okay, it's really thin, and it's really long. It's around 1 meter long, a little bit more. And this is, its basic structure. So we have here the nucleus. The nucleus contains the DNA. We have the cell body, which is the space surrounding the nucleus. And we have this tree, which we call dendrites. These are components that interact with the previous cell in the network. So the nerve is really, really long. Okay, and this is the long part. It's called an axon. It gets all the way to your toe. And then we have this other branches, okay, which are connecting to the next cell. So these branches connect with the previous cell, and these connect with the next cell in the row. Okay, so they can connect with more than one cell, of course. Is that clear?
What happens if people lost their library, like lost an organ or something? I mean, it can still function properly, or?
So it depends. Nerves, generally speaking, nerves are not dividing. We know that some are. We know that there are some stem cells in the brain, which are cells that can differentiate to become neurons. But mostly they're not dividing, so they will have some permanent damage usually. Okay. So this is a picture of Antony van Leeuwenhoek. This is the first microscopist that actually looked at cells. He called them animalcules for animal and molecules. And he looked at different bacteria in different puddles. Okay. So this is Robert Hooke, another English microscopist that also defined the cells as celluli, little rooms, cell. Okay. And this is a picture of the human skin. This is how it looks like. So the DNA, the nucleus, is stained in blue here, and the structures inside the cells, the basic structures inside the cells called intermediate filaments, are stained in green here. Okay, so this is a fluorescent picture of the skin.
What is the orange?
Probably some protein that interacts with other cells, but I don't know.
The colors are because of the equipment, or?
No, when we stain cells fluorescently, we use antibodies that are bound to some fluorescent dye. So this way we can look at it. Okay, this is another picture of fluorescent microscopy. And this is a cell in the middle of division. So you see here what is called centromeres. This is the structure where all the genetic material organizes before it is split to two cells. Okay? It's not really important, the names. It's just to illustrate a little bit about what we're talking about. Okay, so we have organelles in the cells, and what they do is to perform various functions. They're all surrounded with a membrane. Okay, this is a fat, a lipid layer that separates it from everything outside of the cell. Inside the cell, we have other different organelles that also are separate from the cytoplasm, the inner liquid in the cell. Okay. Okay. So this is a cross-section of an animal cell. Here you see the nucleus. The DNA is located right here. This is the endoplasmic reticulum where proteins are made. This is mitochondria where energy is made. And we'll talk more about them, I think, in the next lesson. Okay. So the nucleus, here you have a definition of each of these organelles, and you're going to use it. So please make sure you understand. So the nucleus is the site of DNA synthesis. The DNA is stored in the nucleus, and it carries the genetic material to make up the cell, the organism, everything. This is the mitochondria. Mitochondrion is a single. Mitochondria are multiple mitochondrions. And this is, as Sebastian told you last class, this is the power plant of the cell. This is where energy is made up. So mitochondria breaks glucose, sugar, that you get from your diet, and turns it to energy. This is the cytoskeleton. So it's the structure that looks like this, and it supports the cell. It holds the cell together, okay? So it has some structure. Each cell has a different structure. Neurons have a very unique structure. Liver cells look more like this, okay? They're just square, but they're very similar. And skin cells are relatively round, for example. The Golgi apparatus, as you can see here, we're not going to talk about it. I don't want to confuse you too much, so I'm focusing on the particles that are actually useful in this course. And we have the rough endoplasmic reticulum. This is it. These are the sites where protein is synthesized, is made. ribosomes are sitting on the rough ER, on the rough endoplasmic reticulum, and they are the particles that actually make the protein. Okay? We also have the smooth endoplasmic reticulum. This is the non-spotted part of the endoplasmic reticulum. Okay? Will not go into details. Okay? And we have the plasma membrane, which separates the cell from everything outside. This is not important.
The membrane is larger?
Yeah, the purple thing. Yeah. Okay. What happened? Okay. So Tay-Sachs disease, this is another neurodegenerative disease, is characterized by harmful quantities of cell membrane. They have too many cell membranes, and this leads to a death of the cells. Okay. And here you have the nucleus, the endoplasmic ER. It's basically the same thing. In Huntington's disease, we have a different dysfunction of the ER. I think we have a break now, right? Let's make a break of 5 minutes, okay? So this is how we do in Givat Ram. So we'll do it this way in this class. Okay, so take 5 minutes and we'll come back. Okay, let's continue. So in Huntington's disease, we have a mutant protein called huntingtin. We'll talk more about it, but it can affect calcium metabolism in cells, and it functions in the ER too. And this is Ada Yonat. Ada Yonat is a Nobel laureate from Weizmann Institute who won the Nobel Prize for discovery of the ribosomes, the site of protein synthesis. I hope you heard of her. Ribosomes are located on the endoplasmic reticulum, on the ER, and they are bound there, and they consist of RNA. RNA, this is a specific RNA called ribosomal RNA. So we'll talk more about RNA, but it's an intermediate between DNA and protein. Okay, the Golgi apparatus, here it is. It receives proteins from the ER, and then it delivers them to their destination. For example, if a protein has to be located on the membrane, outside the membrane, for example, a receptor, okay, this is the organelle that sends it out. Okay, sometimes it modifies the protein a little bit before it sends it. And here is a more in-depth illustration of the Golgi. So this is the ER. It sends proteins to the Golgi, and the Golgi has this particles that has the protein in them. Okay, and for example, it sends them outside the cells. These particles combine with the plasma membrane and gets the protein out. So many different degenerative diseases like Alzheimer's and Creutzfeldt-Jakob, which is the mad cow disease, and SMA and Parkinson's and other diseases are characterized by problems in the Golgi apparatus. Okay, so this is why it's important to know a little bit about these organelles, because dysfunction in them results many times in neurodegeneration. Okay, so these particles that the Golgi sends out are called lysosomes. So lysosomes have multiple functions. For example, they get out some materials outside of the cell. They can also function as the trash can of the cell. So if proteins has to be destroyed, they many times get into lysosomes. And in Button's disease, another neurodegenerative disease, we have lysosomal dysfunction. But many other neurodegeneration related diseases are also associated with the same thing. Okay. Yes.
The Golgi is a type of lysosome, or
No. The Golgi is an organelle, and it produces lysosomes. Okay. So a little bit about the cytoskeleton. Cytoskeleton is a skeleton of the cell. Okay. We have different types of cytoskeleton. For example, here you see stainings of these fibers. This is an actin fiber. It's relatively small. Okay. It is very, very abundant. We have intermediate filaments that are stained here in red, and large cytoskeletal units called microtubules. They are made up of a protein called tubulin, and they are very large, 25 nanometers. They're also very long, of course. Okay, so in Alzheimer's disease and corticobasal degeneration, another neurodegenerative diseases, we have microtubule-associated protein. So it's associated to the microtubules. These are the microtubules. Okay, and it is found in the axons, in the extensions of the cell, in the long part of the neuron. Okay, and sometimes they stop working properly, and this results in a disease. By the way, this is a picture of a human brain after he died. And here you see the step here. Okay, so this is a degeneration of this area. This is the frontal cortex in this case. Okay, and this is how a sick brain looks like. This is an MRI picture of, I think it's a different one, I'm not sure. Okay, and here you see these weird holes, okay, right here. They're not supposed to be here. Okay, so a bit more about the cytoskeleton. We have 3 different types, as we said. We have the microtubules, the large ones, the blue ones here, the intermediate filaments, the green ones in this case, and the microfilaments, which are actin filaments. They are the same thing. Okay, and they hold the structure of the cell. This is another staining of some microtubules. Okay, so these are microfilaments, actin. These are the proteins that make the structure of the cell. They hold it in place. Okay, so it also helps the cell to move. This is really important because once a cell has to move, the actin is, sorry about that. Where were we? Okay, so the actin can dissociate in one part, and we have more monomers, more subunits that are growing in the other end of the actin. And this way, the cell is able to move. Okay? What about the nucleus? So, as I told you, this is the site where DNA is, where our general genetic information is. DNA is very compact, and it makes up a structure that is called chromatin. I think I have a slide about it. But the nucleus has inside another small nucleus. It's called nucleolus. It's not really important in this case. But what's important is that we have these holes in the nucleus. They are called pores, nuclear pores. And these holes allow RNA, the messenger between DNA and protein, to get out of the nucleus. Is that clear? You look really confused, so please ask. Okay. So we have these holes. This is how it looks like when you take a real picture of that. This is electron microscopy. Okay, and this is a picture of the same thing. We have these holes, and the RNA gets out of the cells through them to the cytoplasm, to the area surrounding the nucleus.
Question.
Yes?
Again, the RNA gets into
So the RNA is the intermediate between DNA and protein. These are the instructions for the cell how to make the protein. We'll talk more about it, I think, in the next class. Okay? Okay, so probably you saw this structure previously. Okay, this is a DNA structure, a very classical double helix. Okay, it was first described by Watson and Crick in 1952, and also a woman that didn't get the Nobel Prize on the credit. Her name was Rosalind Franklin. She was Jewish, actually, and they used her data to find the structure of the DNA, but never gave her the credit. So this is the double helix, and it has to be really, really compact because it's really long. We have a lot of genetic information in our cells, and somehow we have to fit it in. So how does it happen? We have, it's really, really long. As I told you, it's around 2 meters in each cell, 2 meters of DNA. It's huge. Okay, and it has to be really, really packed. So we have different structures of proteins that help to make it really compact. An example is the histones. These are the yellow things around here. And the nucleosomes. These are the orange, I think it's orange, red, whatever. These are the balls. And around each ball, we have 2 loops of DNA. And the DNA already gets compact before. Okay, so you see this is getting thinner and thinner and more compact. And eventually, it gets to a chromosome. This is the most compact structure of the DNA. And this is how it really looks like. So we have, how many chromosomes do we have, you know? 46. 46. And what happens when something goes wrong? For example, Down disease happens when we have 3 pairs of chromosome 21, for example. Okay, so when something goes wrong when the cell divides, it can result in different diseases like Down syndrome and many, many others. Okay, so these are the 46 chromosomes. These are for the human, by the way. For mouse and for plants or for any other organism, each one has a different number of chromosomes. And you would expect that the human would have the highest amount of chromosome, but this is not true. Okay, I think I have a slide about it soon, but this is the human Y chromosome. So only males have this chromosome because females have 2 X chromosomes. And this is a chromatin. So chromatin is DNA plus proteins. This is the definition. Okay, it's relatively simple, but for this class, it's enough.
Question.
Yes.
The last
Slide?
Yeah, slide.
Yeah.
What is a histone?
Oh. So a histone is a protein that is really, really complicated. But bottom line, what it does is to allow the DNA to be really compact or release it when needed. So, for example, the cell wants to transcribe some protein now because you're really cold and this protein functions in heating the cell. So the histone releases this area of the DNA that has to be transcribed, that has to be expressed, okay? And then it gets back together after the protein is made. Okay, so it opens and closes the DNA structure. Okay, this is another picture of a chromosome. And here you see these lines, the small threads. These are DNA, okay, threads. Here, it's like hairs. Looks like hairs in this case. So this is a chromosome, a typical appearance of a chromosome. And we have these 4 arms. Each of these arms is called chromatid. Okay, and you don't have to remember all these names, okay? So don't be stressed about it. You have the presentations, you can always go back. So before the cell divides, the DNA organizes very, very nicely in structure. All the chromosomes are aligned together in the middle of the cell, and then they split to 2 cells. Okay, So every chromosome has, it's a pair of chromosomes. So you have 2 chromosomes of each kind, one from your mother and one from your father. Okay, and you get different genes in different chromosomes. For example, this chromosome can have a gene for dark hair and the other one for light hair. One you got from your mother, one you got from your father. And one will express and the other won't. Okay, so we'll talk about inheritance. This is more about dominant genes and other characteristics. Okay, so we'll move to the next organelle, the mitochondria. So this is, questions? Okay, so the mitochondria, this is the place where energy is made in the cell. Okay. So it's a fuel. It's the fuel of the cell. It is made up of sugars that are break down. So it can be made of sugars that you consume for your diet. And if there are no sugars available, it is made of fat. Fat can break down and make energy. Okay? And there are other stores of energy in the muscle, for example. Okay. So in ALS, as you remember, the disease that is characterized by lack of movement. Okay, so it's degeneration of motor neurons in the spinal cord. And in the inherited version of this disease, we have a mutant protein that is called SOD1. Okay, SOD1, it is not responsible for many cases of the disease, but this is like one of the most studied genes because we don't really know other genes that do the same thing. And we know that when SOD1 is produced in the wrong way, it's mutant, it is stuck in the mitochondria. It gets stuck there. Okay, and then it gets, there gets more and more SOD1 in the mitochondria, and eventually it cannot function and the cell dies because it cannot produce any energy. Okay, a little bit about another organelle in the cell, the ubiquitin proteasome, also known as the UPS system. Okay, so Sebastian told you a little bit about it in the previous class, but generally speaking, the ubiquitin proteasome is where proteins are broken down. Okay, so when a protein is getting too old, for example, and the cell sees that it is not functioning very well, it tags it in a molecule called ubiquitin. This is the tag. Okay, so it tags more and more and more, and eventually it gets to a threshold that tells the cell, okay, this protein has to be destructed, or as we call it, degraded. Degradation is destruction of anything. In this case, it's a protein. So it gets into this structure, okay, the ubiquitin proteasome, and here it breaks down to single units. Okay, so we have this protein, it unfolds, get in, and break down. It is very, very important for healthy cells, for the health of the cells, that the UPS system works properly. In Huntington disease and also in Parkinson's disease, we have proteins that are not going through the UPS system as they should. For example, in Parkinson's, we have a protein called alpha-synuclein, and it aggregates, it makes clusters in the UPS system, and then nothing can break down, and the cell dies. Any question about the UPS system? Okay. So this is a summary for you of the organelles and what they do. You can just always go back or Google it. Okay. So this is the plasma membrane, the membrane surrounding the cells. This is the inside of the cell. What are the yellow things? Skeleton, yeah. This is the cytoskeleton. This is the outside of the cell. Okay, so sometimes we have to allow things get in and out of the cell. For example, from the bloodstream. So you eat a sugar, the cell needs this sugar, it has to get in. How does it do that? So we have different proteins and lipids that are sticking out of the cell and sensing different materials. When they bind to a specific material, and they're really, really specific, they open like a gate, and I'll show you soon how it looks like, and allow the component to get in the cell. Okay, for example, the sugar or the lipid or whatever it is.
So they don't absorb, they just open it.
Yeah. Okay, so this is a carrier protein. This is how it looks like. So here we have the lipid bilayer, a double layer. This part is made of fat, of lipids, and this fat, and this part is not fat, it allows water to pass through. And we have this glucose. Glucose is sugar, carrier protein. And in this case, we have the glucose molecules, the sugars from your diet. Okay, so the glucose enters to the cells through this protein. It has a specific binding site that fits only the glucose. Other molecules cannot get in. Each molecule has a different structure. So this gate, this protein, is able to differentiate between them. And once the glucose molecule is entering in this protein, in this carrier protein, it changes its conformation and allows it to get out only from a single side.
What would the other one just opt out or something?
Other what?
If not glucose gets in something else.
So you have different proteins for any molecule you need. Okay, so you have a different gate, and imagine one would bind to a circle, one would bind only a triangle. Basically, it's very close to the reality. Yeah. Yes.
Those proteins are not enzymes?
Those are enzymes, but so enzymes need energy to function. In this case, some carriers, some proteins need energy to do that, and some don't. Some work according to the diffusion, according to the concentration. So if you have a high concentration outside the cells, they will allow it to get in, whatever the material is, until it's the same concentration. Okay, so it depends. Okay, and here we see the glucose inside the cell. Okay, and once the glucose is in the cell, there is another conformational change. The carrier protein is now available for another molecule of glucose. Okay, and each time we have many of these proteins in our membranes, and they all function together, but each one is specific for a certain material. Okay, a certain chemical. Yes?
Also, each one of them is going to only one direction, right?
Not all of them. Some go to both directions. This is a simple example, but in biology, everything can get more complex. Okay, so here is a more complex example. So these are ion channels. What are ions? Ions are molecules that have some charge on them. So they have a minus charge, a negative charge, or a positive charge, plus on them. And why do we need these ions in our cells? Any ideas?
Well, the human body also operates on electrical currents.
Right. So
That means you're going to have charges going through the body, so you need to be able to deal with them.
exactly. So neurons are nerve cells. They need these ions in order to function. They function using electrical charge. This is how they send a message in the neuron. And we need these ions to make it work. So we have different gates for these ions. And each one looks a little bit differently and has a different function. We have voltage-gated channel. These are channels that open only when there is a lot of positive charge outside of the cell, and inside the cell it's very negative charged. This is the only case when this gate will open. Okay? We also have ligand-gated channels. We have extracellular, which means outside of the cell, or intracellular, inside the cell. So when will this gate open? Only when these red things will bind to it. So these red things can be different cofactors. So different chemicals that bind and signal this gate to open. We also have mechanically gated channels. So these are channels that open when there's pressure upon them. Is this clear? So you see the picture gets more and more complex. This is a movie. I'm not sure it will work here, but you can watch it. Yeah, it won't work. It's a movie about the cells. So it summarizes everything we were talking about in the last 1 and almost a half hours. You can go over it if you wish, but if you have any questions, I'm here. If not, you're free to go.